Coming to our next chapter, biological oxidation. So here the actual concept of biological oxidation is, so whatever the food we take, it undergoes three stages of metabolism. So primary metabolism is digestion of the molecules, macromolecules. So whatever the carbohydrates, proteins and fats you take in the diet, those are subjected to digestion and they are converted to their simpler forms. So that is primary metabolism. Coming to secondary metabolism. So in this secondary metabolism, so after the conversion of the complex molecules into simpler molecules, they are subjected to catabolism. In the catabolism, the energy content in those molecules is derived in the form of reducing equivalents. So we have the third type of metabolism in the tertiary metabolism. Third type of metabolism, what happens there? In the tertiary metabolism, whatever the energy reducing equivalents was produced in the secondary metabolism, they are converted from the energy from the reducing equivalents is derived in the tertiary metabolism. Right? So these are the three types of metabolism: primary metabolism, secondary metabolism, tertiary metabolism. So in which energy content of the foods is derived is trapped in the form of ATP. So high energy compounds. What are high energy compounds? So based on the energy released by a compound, we can classify them into high energy compounds and low energy compounds. So what are high energy compounds? When the energy released by a substance is more than 7 kilocalories per mole, such compounds we call them as high energy compounds. For example, you can take ATP, adenosine triphosphate, right? And other examples are phosphoenol pyruvate, creatine phosphate. So those are the some important examples for the high energy compounds, right? And when energy released by a substance or molecule is less than 7 kilocalories per mole, such type of compounds we call them as low energy compounds, right? Like glucose 1-phosphate, glucose 6-phosphate, fructose 6-phosphate. All these are the compounds which have energy, which can release energy less than 7 kilocalories per mole. So this is the classification of compounds into high energy and low energy based on the amount of energy released by them. Right. So here the basis is 7 kilocalories. So whatever the compounds which release energy more than 7, we call them as high energy compounds. If it is less than 7 kilocalories per mole, we call them as low energy compounds. Coming to, you should know about what is oxidation, what is reduction and redox potential. So when you are going through this biological oxidation and electron transport chain. So what is oxidation? Oxidation is nothing but loss of electrons is called as oxidation. And coming to reduction, gain of electrons is called as reduction, and redox potential. So when you take in oxidation reduction reactions, so the electrons are transferred from a more negative redox potential to a more positive redox potential. So when you say negative redox potential, so that compound which has negative redox potential can donate electrons easily, right? When a compound has positive redox potential, it has a strong affinity for accepting the electrons. So which can donate electrons easily has negative redox potential, and which accepts the electrons has the positive redox potential. So here, so this is about the oxidation, reduction and redox potential. Coming to electron transport chain. What is electron transport chain? So this occurs in the mitochondria. Mitochondria has outer membrane, inner membrane, intermembrane space, and inside the inner membrane it has a matrix called as mitochondrial matrix. So this inner membrane has the electron carriers and the sequence of electron carriers which are responsible for the transfer of electrons from one component to other component. So this sequence of electron carriers is called as electron transport chain. So these electron carriers are responsible for the transfer of electrons. So those electrons are transferred from a high negative redox potential to positive redox potential, right? So where this electron transport chain occurs? This electron transport chain is located in the mitochondria, so inner mitochondrial membrane. Coming to sources of electrons. So as we discussed in case of secondary metabolism, there the macromolecules which are converted into simpler ones by dilution, they undergo catabolism inside the cells. That catabolism and different oxidations generates reducing equivalents in the form of NADH and FADH2. So those reducing equivalents, they have electrons. Those electrons are the sources for the electron transport chain. So the electron source is the reduced form of coenzymes, that is NADH and FADH2, which are obtained in the various oxidation pathways like beta oxidation, glycolysis, citric acid cycle, oxidation of amino acids. So all those oxidations, they generate the reducing equivalents. So those reducing equivalents are the source for the electrons in the electron transport chain. Coming to components of electron transport chain. So we discussed that there are series of electron carriers in the electron transport chain, and those series of electron carriers, nothing but they are enzyme complexes, they are enzymes. So those enzymes, they have different components, like when you see the most important component is nicotinamide nucleotides. Like, so when you see complex 1, it has NADH. So NADH is nothing but the coenzyme form of niacin, right? So this is nicotinamide nucleotide. So NAD, nicotinamide dinucleotide is the coenzyme that is present in the complex 1. Coming to flavoproteins. So any enzyme which has FMN as a prosthetic group, such type of proteins we call them as flavoproteins. The complex 1, NADH coenzyme Q reductase, is nothing but a flavoprotein because it has FMN in its structure. Coming to iron sulfur proteins. The role of iron sulfur proteins. So iron sulfur protein is nothing but it has iron and sulfur complexes. Those iron sulfur proteins, they exist in two states, oxidized state and reduced state. So iron exists in two forms, oxidized state and reduced state. So those are responsible for the transfer of electrons. Right. Coming to coenzyme Q, so this is the mobile component of electron transport chain. So when you see all the remaining components, they are fixed, and coenzyme Q is a mobile component. So it can move from complex 1 and complex 2 to the complex 3. So it carries electrons from complex 1 and complex 2 to the complex 3. So it is a mobile component. Right. It is also called as ubiquinone. So coenzyme Q or ubiquinone. Coming to other component, we have cytochromes. So cytochromes are nothing but a conjugated proteins having the prosthetic group heme. So heme is a porphyrin ring with the iron in its center. So porphyrin rings are nothing but they have 4 pyrrole rings. So 4 pyrrole rings form porphyrin ring, and when you incorporate iron into that porphyrin ring, it becomes a heme. So heme plus the protein part becomes the cytochromes. Right? So that's about the cytochromes. So these cytochromes are the very important components of the electron transport chain. So they are the very important components of complex 3 and complex 4. And coming to the flow of electrons, how the electrons will flow. The electrons will flow from complex 1 to the complex 4 and finally to the oxygen, which accepts those electrons and is reduced to water. So when you see, so NADH donates its electrons, transfers its electrons to the complex 1. So once it transfers its electrons to the complex 1, it is oxidized. So actually NADH is a reduced form of the coenzyme niacin, coenzyme form of NAD+. So NADH is reduced form. That NADH in its reduced form donates the electrons to the complex 1. So when it donates, it is oxidized. Right? So now complex 1 transfers the electrons to the coenzyme Q. So right, and same way FADH2 succinate dehydrogenase is the reaction in citric acid cycle which catalyzes the conversion of succinate to fumarate. There FADH2 is generated, and in case of beta oxidation also FADH2 is generated. Wherever the reactions in which FADH2 is generated, that enters into that donates the electrons to the electron transport chain through the complex 2. So NADH donates electrons to the complex electron transport chain through complex 1, and FADH2 donates electrons, transfers electrons to the electron transport chain through the complex 2. Right? So when electrons are transferred to the complex 2, those are again carried by the coenzyme Q to the complex 3. So electrons from complex 1 and complex 2, they are transferred to the complex 3 by a mobile component called as coenzyme Q. So coenzyme Q, when it accepts the electrons, now it will be reduced. So actually it is in oxidized form, but when it accepts the electrons, it is reduced to reduced form of coenzyme, right? Coenzyme Q. So coenzyme Q, now having the electrons, now it reaches to the complex 3 and transfers the electrons to the complex 3. In complex 3 we have different components. It has iron sulfur proteins, cytochrome B and cytochrome C1. So that iron sulfur exists in oxidized form. Once it accepts the electrons, it will be reduced to reduced form of iron sulfur, and that reduced form of iron sulfur protein transfers electrons to the cytochrome B oxidized form, that will be reduced to cytochrome B reduced form, and cytochrome B, once it is reduced, transfers electrons to the cytochrome C1 oxidized form of cytochrome C1. So once it accepts the electrons, cytochrome C1 is converted to reduced C1. So that reduced C1 now transfers electrons to the cytochrome C. So we can compare the function of cytochrome C with that of coenzyme Q. That is, coenzyme Q function is it transfers electrons, carries electrons from complex 1 and complex 2 to the complex 3. Same way, cytochrome C function is transfers electrons from complex 3 to the complex 4. Right. So complex 1 is NADH coenzyme Q reductase, complex 2 is succinate coenzyme Q reductase, and complex 3 is cytochrome C reductase. So these are the names of the complexes, complex 1, 2, 3. Now from the complex 3 cytochrome C reductase, the electrons are transferred to the complex 4. So those are carried by the cytochrome C. So that cytochrome C transfers the electrons to the complex 4. In this complex 4 is nothing but a cytochrome oxidase. Cytochrome A, A3 are the components of the complex 4. So cytochrome oxidase is the complex 4. Now cytochrome oxidase complex 3 having cytochrome A, A3 in oxidized form, once it accepts the electrons from the cytochrome C, it is reduced to cytochrome A3. So that reduced cytochrome A3 transfers the electrons to the final acceptor of the electrons, that is oxygen. So those electrons now transfer to the oxygen, results in the reduction of oxygen to water. So the electrons from the NADH reduced form of niacin, that is NAD plus, NADH and FADH2, they are transferred to the complex 1 and complex 2, and they will flow from the complex 1 and complex to the complex 4. So finally from complex 4 they are accepted by the oxygen, and that is got reduced to water. So when the electrons are flowing from complex 1 to complex 4, so the energy released during the transfer of electrons through these complexes is responsible for the pumping of protons from mitochondrial matrix into the intermembrane space. So as we discussed, there is outer membrane and inner membrane, and the space between outer membrane and inner membrane is called as the intermembrane space. So the protons are pumped into the intermembrane space through the complex 1, complex 3 and complex 4. So these complex 1, 3 and 4 are called as proton pumps, because protons are pumped from the matrix to the intermembrane space through these complexes. Right. So the energy which is released during the transfer of electrons from these complexes is responsible for the transfer of pumping of protons from the matrix to the intermembrane space. Right. So when protons are pumped into the intermembrane space, that creates a gradient called as proton gradient. So that proton gradients, once generated, so those protons in the intermembrane space, they again try to come back into the matrix. But they can go into the intermembrane space, they can be pumped into the intermembrane space from proton channels like complex 1, 3 and complex 4 proton pumps. But they cannot re-enter into the matrix through these proton pumps. But the site of entry is different. So they enter into the intermembrane space in one way, and they again re-enter into the matrix through another way. So that another way of entry for the protons is ATP synthase, also called as complex 5. So that ATP synthase function is So it allows the entry of protons which are accumulated in the intermembrane space into the matrix. So that those protons which are accumulated are pumped into intermembrane space, they again come back into the matrix through that channel called as ATP synthesis. So that ATP synthesis has two subunits, FO subunit and F1 subunit. So those protons, they enter into the matrix through FO subunits. So they enter through the FO subunit. So when they are passing through the FO subunit, that creates a conformational change in the F1 subunit. So that F1 subunit, which now undergoes some conformational changes because of entry of protons through that FO subunit. So those conformational changes in the F1 subunit catalyzes the synthesis of ATP. So creation of phosphoanhydride bond is because of the conformational changes in the ATP synthesis that is brought by entry of H plus ions or protons into the matrix from the intermembrane space. So that is in the synthesis of ATP. So actually this is a coupled process. That's why the ATP synthesis in mitochondria is called as oxidative phosphorylation. So there is oxidation of reducing equivalents, and that is results in the creation of a proton gradient, and that proton gradient is responsible for the synthesis of ATP. So the oxidation is coupled with the phosphorylation. So that's why synthesis of ATP in mitochondria we call it as oxidative phosphorylation. So ATP is synthesized in two ways, that is main way is oxidative phosphorylation and the other one is substrate level phosphorylation. So in substrate level phosphorylation, this ATP is synthesized directly from the substrate, so that's why we call it as substrate level phosphorylation. So it won't require the help of this electron transport chain, or it won't require the help of the reduced coenzyme forms like NADH, FADH2. Then there is no need for the entry of those, creation of those reducing equivalents and their entry into the mitochondrial matrix, and for the transfer of electrons from the complex 1 to complex 4 that creates a proton gradient, and finally ATP synthesis. So this whole mechanism is not required in case of substrate level phosphorylation. Simply ATP is synthesized directly from the substrate. So that is the minor way in which ATPs are produced, but the major mechanism in which ATPs are synthesized is oxidative phosphorylation. So there is one theory which proposes this mechanism that is called as chemiosmotic theory. So chemiosmotic theory says that ATP is synthesized because of the proton gradients. So when electrons are flowing from the complex 1 to the complex 4, so that creates a proton gradient. So that proton gradient is responsible for the synthesis of ATP. So that is what the chemiosmotic theory tells us about synthesis of ATP by oxidative phosphorylation. Right. So mechanism of oxidative phosphorylation, till now we have discussed about that only. Mechanism, how oxidative phosphorylation occurs, it's simply by loss of electrons from the complex, from the NADH and FADH2, and that those electrons are transferred to the complex 4. So that process we call it as oxidation, and phosphorylation is when they enter the protons when enter into the matrix. So that is responsible for the synthesis of ATP. That process we call it as phosphorylation. Right? So there are some inhibitors of the electron transport chain. Right? So there are some inhibitors which inhibit complex 1, complex 2, complex 3 and complex 4. When you see complex 1 inhibitor rotenone, insecticide rotenone can inhibit the complex 1, and complex 2 is inhibited by carboxyl, and complex 3 is inhibited by an inhibitor called as BAL, British anti-leucite, and complex 4 is inhibited by cyanide, carbon monoxide, hydrogen sulfide. So these are the important inhibitors which inhibit complex 4. Right? So they prevent the oxidative phosphorylation. Coming to uncouplers of oxidative phosphorylation. What are uncouplers? So oxidation and phosphorylation is a coupled process, but these compounds, substances called as uncouplers, the function of these uncouplers is they prevent the proton gradient creation. Right? So generally oxidative phosphorylation is a coupled process. Oxidation occurs, synthesis of ATP occurs. So that synthesis of ATP is because of proton gradient. Here oxidation occurs without creation of a proton gradient. Protons will be pumped into the intermembrane space, but those protons again they come back into the mitochondrial matrix. So not only H plus ions, other positive ions also they enter into the mitochondrial matrix through the inner membrane, because these uncouplers substances they change the membrane permeability, inner membrane permeability, such that the positive ions they can freely enter into the matrix through the inner membrane space. So actually that won't occur in general mechanism, but whenever uncouplers are there, those uncouplers they allow oxidation and they break that, uncouple that phosphorylation. So uncoupling of oxidative phosphorylation occurs here by the action of uncouplers. And example you can say oligomycin is a uncoupler. Apart from that, there are two physiological uncouplers like thyroxine and thermogenin. So those two are responsible for the uncoupling, that is they allow oxidation without phosphorylation. So whatever the energy released, it is released as heat without generation of ATP. So that is a mechanism involved in the uncouplers. Thank you.